Hey guys, this is Kendall Terry, and this is Animal Development Part 2. So let's jump right in. So we left off with gastrulation on the last video, so now we're looking at what is organogenesis. Organogenesis is really just the creation of the organs. And this involves a lot of things. This involves folds, splits, clustering of cells. It involves stuff sometimes with the ectoderm, sometimes with the mesoderm, sometimes with the endoderm, and we're going to look at each one of those and what they actually go to develop in the system. But really, at this point in development, we're at a point that you're looking at the organism really starting to look like and act like and become the organism, the adult organism that it's going to be. So let's look at that. So early organogenesis in the frog and the embryo, primarily we look at the neural tube forming, and you can see that here. You've got the neural fold that develops, and this is pretty fascinating. If you can watch a video of this, you should, of the embryo in development, and this neural fold will kind of like form in the middle of that disc. For mammals, it's a disc. For other organisms, it has different shapes, as you can see in the picture here of the frog. But there will start to be this fold that will develop, and you can see that right here on the screen. It starts in the ectoderm, and in the ectoderm it will start to fold, and you can see that here. You get the neural fold that will develop in the neural plate, and then eventually that neural fold will pinch completely off to where you'll form down here a neural tube that will be pretty much the precursor to the spinal cord in mammals. And then the neural crest, and the neural crest will go on to become other structures in the development of that embryo. Now, there's a couple things that I want to point out since we're on this slide here. One is that the notocord is present. The notocord is one of the things that's present in all chordates, and in most of the more advanced chordates, this will become other things. Maybe it'll become part of the discs in the spinal column, whatever may be the case. In most organisms, in early vertebrates, this structure will actually stay present in adults, and it becomes a point of attachment for things like muscle in that organism, and it gives some flexibility. It's not rigid so that they can't move. So you think about early chordates living out, maybe we're talking about fish in an ocean, they don't have the hard exoskeletons of maybe a primitive ancestor, but they still have that point of attachment for muscle and stuff. So they've got the point of attachment, but they're more flexible in their movement than a maybe crab or something like that with that hard exoskeleton. So this was an early development point for the vertebrate, was the notocord, and you can see that right here. It's made up of the mesoderm. The other thing I want you to see in this picture, and it's kind of hard to see in the bottom one, but in the top one here, the cross section of the early embryo, frog embryo, you'll see the somites form. The somites are early bone structure. In this case, it would have been the vertebral column, but what you see here is that those somites, whenever that neural tube is developed, those somites are there as well, and those will be those early muscle, like the vertebrae and skeletal muscles in that area. So if we want to look at those three embryonic germ layers in vertebrates and what they develop into, this breaks it down for you. I'm going to quickly run through these. So if you look at the ectoderm, you're talking about the epidermis of the skin and its derivatives. That includes your sweat glands, hair follicles, things like that. The epithelial lining of the mouth and the rectum, the sense receptors of the epidermis, the cornea and lens of the eye, the nervous system, the adrenal medulla, tooth enamel, and epithelium or pineal and pituitary glands. So you see it's got kind of a diverse group. When you think about, that's the outside covering of that embryo when it's developing, and then it develops, of course, the skin and that sort of thing makes sense, but then you throw in there stuff like the nervous system, and because of that neural fold that happens and it pinching off, that's how the ectoderm becomes that. That's a little interesting to think about that, though, maybe. Then you've got the mesoderm here. The mesoderm is remember when that was forming in the blastosphere, or the blastopore, that was the space that develops between the endoderm and the ectoderm, that inside cell area. That's the mesoderm. And this is stuff like the notocord, the skeletal system, muscular system, muscle layer of the stomach and the intestines, excretory system, circulatory and lymphatic system, reproductive system, except for the germ cells, the dermis of the skin, lining of the body cavity, adrenal cortex, and then the endoderm, which was that inside layer that came in. Primarily that consists of a lot of times the gut tube, and if you look, that's the epithelial lining of the digestive tract, the epithelial lining of the respiratory system, the lining of the urethra, urinary bladder, and reproductive system, liver, pancreas, thymus, thyroid, and parathyroid glands. A lot of that related to that gut tube type area there. One thing I want to touch on real quick is the 4 extra-embryonic membranes of the amniotic egg. This is kind of, when you think of this, a lot of times you think of maybe a chicken egg type deal. That's probably what you're most familiar with. The 4 main extra-embryonic membranes are the amnion, the Allantois, the Chorion, and then the yolk sac. So when you look at that here, you can see those areas that are highlighted, and you might not be able to read those words, but you can go to your book and use that as a resource to help understand all of these. I'm going to just go through them real quick. The amnion protects the embryo in a fluid-filled cavity that prevents dehydration and cushions mechanical shock. The Allantois functions as a disposable sac, and this will be what kind of picks up the metabolic waste during development. The yolk sac, which contains the nutrients, a stockpile of nutrients for the egg, and then the Chorion, which is the membrane of the Allantois, and it helps exchange gases. That's its job. I got mixed up down here in the reading. It helps exchange gases between the embryo and surrounding air. Oxygen has to come in because cellular respiration is going on, and carbon dioxide has to go out. And we think about like a chicken egg, you don't think about the fact that oxygen is going in and out of that egg shell. The shell itself is able to exchange carbon dioxide and oxygen without any problem, and then it continues to go through this membrane to get to those cells during that development. Now in us, we don't have to worry about that kind of exchange there, but that exchange is going on in the placenta as mom's blood comes up to a point, and then baby's blood to a point, and they're exchanging nutrients, oxygen, carbon dioxide, things like that through that point in the placenta. So it's just a different thing to look at. You need to understand the embryonic membranes and what they do. So when you look at some key stages of development, this is looking at animals to plants. A little comparison here. We've already talked about some plant stuff in the past. Some of this is a little hard to see probably in the picture. It's a little light. Sorry about that. You've got early cell division from the zygote to the 8-cell blastopore to the blastula to eventually then gastrulation, where you'll have the influx there that creates the gut, and then eventually organogenesis, the development of the actual animal. In plants, you have the zygote that will go through a 2-cell division, and then from that 2-cell division, you'll get this embryo develop. One of those will keep as an anchor point in the seed. The other then actually develops into the embryo, and in that embryo stage inside of that seed, you'll have both a root apical meristem and a shoot apical meristem. These will be the first areas when that seed starts to germinate to get out into the soil, roots to absorb to absorb water, and then the shoot up high, get those leaves out quick to start doing photosynthesis so that you can start growing taller faster. So there's a little difference in the development, enough to start to understand some of the differences, compare them, but also looking for what are any similarities that might be there in that development.